The topic of today's discussion is stem cell therapy as a potential treatment for long COVID. Throughout the presentation today, we're also going to cover not only findings of studies in long COVID, but also the potential and the implications for future studies in ME, CFS. So I'm really delighted to have with us to have this conversation, not only an expert in this field, but also an inspiring leader. Is Donna Chang. Donna is the founder of Hope Biosciences Research Foundation, which is based in Texas, in Sugar Land. She's a graduate of University of Toronto and Johns Hopkins University. And in addition to having a depth of understanding of cell therapy and in that field, but also really the expertise in bringing this forward as really as a potential treatment option. She's also a happy mother of school-age twins, which I'm sure is a challenge in itself, and really brings this expertise with a lot of compassion for our community. So really excited to have you with us today, Donna. And let's kick it off.
Well, wonderful to be here. I'm so thrilled to just be able to spend time with you all today and kind of give you an update on where we are in terms of our research and where we see the potential for this research to go. Just a couple of things before we get started. I know that patients these days are a different kind of breed. A lot of us are doing a lot of our own research with increased resources. And so we have a population that knows a lot and knows a lot of details and those that don't. And so what I'm trying to do is I'm going to present based on, the assumption that we just don't know anything about this and about stem cells in general. I'm going to just speak very generally. And I can answer all of the molecular biology questions that you all might have, and we can maybe do that in a different way. We have put together some resources that can help answer specific mechanistic scientific questions. For this presentation, we're going to, again, keep it to a general audience. And hopefully I can answer many of your burning questions through this. So I'm going to walk through this as logically as I can. And first, I think we're going to start off with, well, what are stem cells? Okay, so stem cells are essentially, the way we can think about this is picture yourself when you were in your mother's womb. So you see that right on the screen. We were at some point, we were embryos forming in our mother's wombs, and we were essentially a big ball of stem cells. Okay. Now, this is terribly unscientific, but I just want you to kind of see what happens as we age. So although we're a big ball of stem cells that end up becoming, so a cell that's a stem cell has no identity, it will end up transforming into maybe skin, maybe your brain tissue, heart, bone. Everything that makes you starts from that initial stem cell, so an embryonic stem cell in this case. But what happens after we're born? So it turns out that after we're born, we still have stem cells in our bodies. And different stem cells have different levels of responsibility. Some are going to create new tissues, some are going to regulate our immune system, some are going to make new blood. There's different functions of these cells. But what we know in general is when you're younger, you have lots of them. Babies are filled with stem cells. I mean, they're growing, actively growing. When you get to a state where you're fully developed, we think that you're at a optimal regenerative capacity. When you're, in your 20s, you're losing lots of cells every day, but you're making them up just as quickly. So your cell turnover rate is super efficient. It's probably the reason why we do a lot of stupid things when we're at that age, because we feel like biologically invincible. And that's exactly what is happening. And then as you begin to get older and older, our regenerative capacity starts to change. So a real clear example of this is, you get a cut on the surface of your skin. You notice that when you're younger, you can heal like that, right? That scar goes away, everything looks great. But the older we get, sometimes that lingers. It takes a lot more longer for bones to heal, for scars to heal. And so that is our regenerative sort of capacity. And what we know is there are cells that are responsible for regeneration. Now, if I were to ask a real basic question, right, which is if there are stem cells that can regenerate our bodies, then how can we replace those cells? Now, it's a simplistic question, but it has a very complicated answer. And the complexity of the answer is why we see generally a pretty just not such an exciting field in stem cell research. I mean, you've heard of many different stem cell applications, particularly in animal studies and proof of concept. And I think we understand that, wow, these cells have lots of potential. But when it comes to actually translating them into a human and translating them into successful, randomized clinical trials, there's a huge gap. Okay, so I can tell you that you can give one infusion of cells to a mouse with pretty much any disease and you can fix it. But that is not going to translate, again, into a human. In order for us to be able to translate these technologies into humans, these three challenges have to be solved. Okay, this is my opinion on this matter. The first is you have to figure out what's the appropriate cell type to figure out to solve your problem. Okay, so if your problem is a vascular problem, then you need some stem cells with angiogenic properties or properties that can actually rebuild blood vessels. If you have a autoimmune disease and you need something that can actually regulate the immune system. So the cell type becomes very important. Then you have to look at the quantity of cells. Just like any medication we take, there is an optimal dose. Just because there's stem cells does not mean that there's no optimal dose. There is, and we also have to be able to quantify that and characterize that, and also make sure that the amount of cells is an appropriate amount that's going to achieve a certain result, right? If you get this many cells, we want to expect a certain result. Right now in cell therapy, that is an extremely obscure topic because we tend to deal with cells that are sometimes fresh, but most often they are frozen. Okay, so they're cryopreserved cells. And when we do this, there's a lot of questions as to what is the quantity that you're actually receiving. So if you freeze cells and you thaw them, then how many are actually alive at the end of the day when you receive them? So that is also within the quantity conversation. And lastly, which is, I think this is the hardest part of all three sort of challenges, is the repeatability aspect. If you think about disease, it's not one size fits all. Okay? You have injury, you have disease, and within disease, there are chronic degenerative diseases, and then there are new onset autoimmune diseases. And depending on the condition that you have, you need to be able to have a treatment that's going to be able to be repeated and safely repeated so that you can solve that problem. A lot of times in clinical research, we tend to forget about this because nobody likes the answer, well, we don't know how many. Nobody likes that. We like to know, okay, 5 doses of this antibiotic is going to fix you. Like, we want to know that, right? And so that's where this becomes challenging. But it's also, if you look at it on the bright side, the fact that you have a safe, repeatable treatment means that you're not so stuck. You're not regimented, and therefore diseases of different stages cannot mainly all be addressed. Okay, so those are the challenges. Now let's go back to the first challenge, which is finding the right cell. Okay, so let me give you a good example of a good source of stem cells. Now, this is where your body makes stem cells. That's probably a good place to look at. This is bone marrow. So bone marrow is where stem cells are made in your body. They're readily made. There are different kinds of stem cells that your body makes. The first are called hematopoietic stem cells. These guys make only blood, red blood cells, white blood cells, platelets, that kind of thing. Now, I'm going to just take a easy example, like Alzheimer's disease. Okay, Alzheimer's disease, our brains are falling apart. We need to rebuild our brains. Are hematopoietic stem cells going to be effective? Probably not. Blood isn't the major issue. So we are looking at a different kind of stem cell. And in the bone marrow, there are these cells called mesenchymal stem cells. Now, these cells are pretty much found in any tissue of your body. And they are what we call multipotent cells. They can pretty much become everything other than blood. But there are also other functions. So I don't want us to box stem cells into cells that transform into tissues. That's just one function. Mesenchymal stem cells have lots of different functions. And these are I pulled just a handful to kind of give you an idea of what they do. And they're incredible cells. The fact that we have these sorts of cells in our bodies is such a powerful tool, because they are, multipotent. Again, they can become lots of different tissues. So, if you need new cartilage or new you need to repair an injury, any of these things, these cells can actually become. But they also prevent cell death. They can make new blood vessels. They're also very powerful in terms of supporting antibacterial function. So we've seen that in sepsis patients. But probably the most important function that they have is their immunomodulatory capacity. So they are constantly regulating our immune system. How? We have answers for that. Again, that's a more technical conversation. But if you can just, I think it's well known in the field that MSCs are excellent at being able to regulate the immune system. So there's lots of applications when you talk about immunity, because immunity in cancer, immunity in diseases like COVID, autoimmune diseases like MECFS, rheumatoid arthritis, and all of the other autoimmune diseases that come along. So that's why we see MSCs as being a focus for us and a lot of other people, by the way. I think in the last year, there have been over 80,000 publications on MSCs. That is a testament to how versatile they are. So now we have this great cell. We know, a few of them can do a lot in animals, but how are we going to make it effective in humans? That's where quantity, again, we have to think about how many of these MSCs are going to be necessary to fix the problem. Well, first, we need to be able to make these quantities, because again, our bodies are, every day that goes by, we have less of these cells. So if you want a technology that can translate across ages and across disease situations. So let me give you an example of, if you have disease present, you've been suffering from disease, your cells are basically working overtime. they're made in the bone marrow, but they're being used up. So if you were to take somebody who's been suffering from disease for a long time and you were to pull their bone marrow, you would find very few cells in there. It's because the cells are all being deployed. So if you're dealing with a sick population, then that means the source that you are working with is you're limited. You don't have enough cells to create a therapeutic difference. So this is why we have to do things in the lab to help with this. So the idea has been, if we're able to take a small amount of tissue or sample from a patient and be able to extract their stem cells and then grow them in a lab, simply culture them to increase the number of them, then perhaps that's how we can increase the quantity. So we at Hope Biosciences is a company that works on multiplication of cells. That's what the expertise is really in. We started the company in 2016 with that objective. I'll talk a little bit more about the foundation as we go into the research conversation, but the technology that I'm about to show you was developed really to meet the requirement of being able to create clinical quantities of cells that are of great quality. So I want to show you, this is a good example of where the industry is today. Okay. If you were to take 1 million stem cells, mesenchymal stem cells, okay, from now on, when I say stem cell, we're talking about your own mesenchymal stem cells. Okay. If we start with 1 million of those cells and we seed it on a flask is essentially a bigger petri dish, and you let them grow, after 5 days, if you look at the right side here, you'll end up with 1.5 million cells. Okay, that's in the industry a really great achievement. Here's the problem. If I were to tell you that 200 million cells is what is going to be required to create a therapeutic dose, then that's a lot of cells. That's a lot more cells that you're going to have to make in a relatively short amount of time. And you have to do it in a short amount of time because the cost of the drug is going to depend on how fast you can do this, how expeditious we can be. So what we do here is we can make 1 million cells and turn them into 32 million cells in just 5 days. And every day after that, there's a population doubling, and we can get to, within a 10-day period, we can achieve a 200 million cell dose. Now, 200 million cells, how do we get there? That's a long story in and of itself. That's another webinar for one day, but that we have established that. That's what we have established with FDA. We have a drug master file on there, and it's standardized. So everybody gets the same dose. That allows us to test the same thing, right? Everyone's going to get the same dose. So now we can actually gear clinical trials to understand what the implication of that dose is, right? Whereas if everyone's getting different doses, it would be very difficult to sort of track. So if you can just believe me that we've figured out the quantity issue, I'm going to now just address repeatability. Now, no matter what disease you're dealing with, there is going to be some repeatability that's needed. We have conducted a clinical trial in rheumatoid arthritis, where rheumatoid arthritis is also an autoimmune disease. And we thought that it would require a few infusions throughout a course of time to be able to sustain the effects. But we published the results to show that actually a single injection, we saw how long can a single injection actually last in terms of symptomatic relief. And it showed that a whole year could go by with good symptomatic relief. So that's groundbreaking in that we're capable of doing that now. But when it comes to reducing inflammation consistently, so changes in the actual blood inflammation, that may require more infusions over time. So In order to make cell therapies repeatable, I ask this question frequently. The question is, would you like your cells fresh or frozen? And I think the natural instinct everyone has is to say, oh, of course we want fresh cells. Okay, so frozen food doesn't taste the same as fresh food. Frozen cells are not the same as fresh cells. Frozen cells are not the same as fresh cells. They simply are not. When you go through a freezing cycle, there are some changes that are going to occur. It's inevitable. And what you're dealt with at the end are going to be cells that we just don't know how many are viable when they're going into the patient. So what we have developed is a way to not use frozen cells at the time of treatment. So we know that what we're giving, if we're giving 200 million cells, that we know all of them are alive. So how we do this is in the autologous trials. So if I were to take a sample of your fat, I would take 10 grams, which is about a tablespoon of fat, would be sent here. And we would isolate just the mesenchymal stem cells out of that 10 grams of fat. That is approximately 100,000 MSCs. That's it, 100,000. Remember, we have to make 200 million per dose. Okay, so what we do is we isolate the MSCs and then we go through the culture process to make lots of cells. And then we harvest them and we bank them and we cryopreserve these cells. Now, this step does require freezing. And now you're probably asking, well, I thought you said the cells are fresh. So this is what we do with those cells. When treatment is scheduled, okay, whether now this is an autologous or whether this is an allogeneic, meaning a donor cell product, 10 days before the patient comes in, we're going to take out a vial of cells and seed on a flask. This is that 1 million cells that I was talking about. We seed and then we go through the culture process. And then 10 days later, we will harvest. This harvested cell product is going to be infused within, there's a 4-day window after QC has passed the product. 4 days, there's a 4-day window in which the patient can receive the cells, thus allowing us to be able to package these up and ship them. If Amazon can ship in a day, it's like under a day these days, but if Amazon can ship like that, we can too. We send cells coast to coast. And we can, from a single tablespoon of fat, we can make over 1,000 doses, 1,037 doses on average. So there's lots of administrations that can be made from a single tablespoon of fat, which now changes the game, because if cells can be freshly made on demand, then now we can really address the difficult questions. Questions like, well, how many cells is it going to take to fix long COVID? How many cells is it going to take to fix Parkinson's? All of them are going to have different answers, and the interval at which we treat are also going to have different answers. But now we have cells at our disposal to be able to measure those things in standardized clinical trials. So I hope that makes sense. In terms of the logic of how we got here. Like, we didn't look at disease first. I hope that makes sense. We're a bunch of cell biologists. And so we said, we just want to address what the issues are when it comes to cell therapy currently in the United States. What are the limitations? And the limitations, as far as we saw, is cells are not fresh. They're not repeatable in any real way. And we're not able to get standardized results, because if you go to phase 3 clinical trials, they all tend to not do so great. So we got to figure out, how do we make sure that everybody can get reliable results? So that's how we got here. So now technology and use, Hope Bio right now proudly serves over 30 FDA approved protocols, expanded access protocols, and phase 2 clinical trials. We have a very large program right now in Parkinson's disease, multiple sclerosis, and now traumatic brain injury. And now long COVID. We've had quite the history with COVID. And, I'll tell a funny story real quick about COVID. After the company was founded and we had developed this technology, now clinical research needed to take place. And so we started the Hope Biosciences Research Foundation to be a foundation that could study diseases, any kind of diseases. And we weren't like commercially driven. The foundation had more liberty to kind of look at whatever unmet need there was. And we started the foundation March 5th, 2020. And for any of you that remember, that was sort of the day the whole world shut down. No one was allowed back in the office. So we said, we got to make ourselves useful. We just started this foundation. What the heck are we supposed to do? So we said, we're going to go into COVID research. We had just gotten approval to do an Alzheimer's study. We scratched it. We said, the priority right now, we don't know if we're going to even make it through COVID. We got to do COVID. And so way before the vaccines got their trials started, we started the first trial to protect ourselves against COVID. Now, the logic was, first, we don't know anything about COVID. 2, we have nothing to protect ourselves. 3, we know that our cells are immunomodulatory, meaning they can regulate the immune system. So we know that if they can regulate the immune system, they can probably protect us against whatever's coming our way. So that was purely the logic that we used to submit our application to FDA, and we got it cleared. Within 30 days, we got it cleared. And so that's how we ended up knee-deep in COVID-19 research since then. We've done trials, very challenging trials in hospital settings. I can tell you, I never wanted to do that again. It's, I still am kind of recovering from that whole experience. Long COVID came along when we were sort of identifying people calling us constantly about this problem. Now, there was no diagnosis for it at the time. Long COVID wasn't even a term. I think we were just collecting it and we were calling it post-COVID-19, syndrome. And we were just collecting calls from people saying that they just can't get over it. They're not positive for COVID anymore, but they can't get over this. And now it's been a year or a year and a half. And a lot of these people had COVID even before the testing was available. So they didn't even know, there was no even confirmed diagnosis to begin with. And we thought, okay, for whatever it's worth, we're going to call this chronic COVID. And we saw pretty much every expression of this condition. Some patients, no 2 patients are exactly the same. They have similar symptoms and we are kind of tracking all the symptoms, but it was a very challenging population, but it looked to us like an autoimmune neurodegenerative vascular condition. I mean, it had components of everything. And so when we kind of looked at, well, if MSCs are great at all of these things from a function standpoint, then I think it could help address multiple areas of long COVID. But you can see the challenge here, right? I cannot tell you one particular pathway in which these cells are going to affect the disease, at least in the disease cycle. And frankly, we don't even understand all there is to understand about the disease itself. So it's hard to make those exact correlations, but if you can just take a step back and look at the function of the cells, I think you can appreciate how they could be useful. And so sure enough, we decided to just do a small pilot. We'll take 10 people who are clearly struggling with long COVID. Most of them had it for at least a year. And we conducted the study. They did receive autologous cells. Okay, this is what we call our HBP COVID-01 protocol. This was the 10-patient pilot. Everyone got cells, so there's no placebo group. It was really for us to determine whether this is worth even looking at. So
and Donna, just to clarify, so these 10 people receive infusions of cells that came from their own fat tissue, right?
correct. This was an autologous study. So each patient, after screening, had gone through a fat extraction. So you see, after screening, there's a window of time where they are able to get a fat extraction. That fat extraction comes to us, and then we go through the banking process, like I showed you. And then after that happens, we're able to generate the 1,000 treatments we're able to make is after banking. So they received 5 infusions, 1 billion cells, 200 million cells. The first and the second infusion were 2 weeks apart, and then the rest were 4 weeks apart. From an interval standpoint, people ask, what was the logic behind the timing? It's really, we like to keep a couple of doses sort of a little bit timed, a little bit closer together, because you're sort of trying to overload the body, like just give the body a lot of cells because it's been depleted. I mean, it's going through a challenge. So we're trying to give them lots at one time, and then space it out in a schedule that will work with people's work lives and, people's lives in general. So we're just trying to create a schedule that'll work, that people will actually finish. In clinical trials, it's very important. If you are thinking about participating, it is critical that you finish, that you go through all of these visits, because, missing data is what, kills trials. That's the side note. Okay, so.
And then one more question. This study was approved by the FDA, and the FDA needs to approve these kind of trials because they view your process as a product. Is that correct?
Yeah, a lot of people ask, well, why is this a drug if it's my own cells?
Right.
And it's a drug because in the United States, if cells are taken outside of your body for longer than a 24-hour period, no matter what's happening, it doesn't really matter. If it's longer than 24 hours, it's been outside of your body, it's going to be a drug. So somebody's going to regulate it. And in this instance, it's going to be FDA. And FDA has said, if it's outside of your body for longer than 24 hours, then it's going to be a drug. And they're going to regulate it. And therefore, it has to go through drug trials, no different than any chemical compound that's out there. You have to go through these regimented trials. Fun fact, well, not so fun, but the whole drug development process, they say that it takes now an average of 12 years to get a drug passed through all of these phases, and can cost now, up to $1 billion. So that's the reality we face. And, when you get to the end, there's even only a 7% success rate. So companies or drugs that go through this pipeline, there's only a 7% that actually get approved at the end of the day. So it's very challenging to do this. And when you're trying to take natural cell products, where if you look at this trial, each patient received their own cells, right? So if you think about that, are my cells and Oved cells the same? Technically, they're different drugs. It's 10 different drugs if you think about it. But what we're trying to do is standardize it. And we're saying that even if these cells are coming from 10 different people who are sick in different ways, we still know that if in the lab we're creating a standardized product and they're all getting the same dose, remember I go back to the quantity of cells. If we do that, then we feel like everybody at the end will get the same drug if you want to make it a drug. So we're trying to make this as standardized as possible. It is challenging when you're dealing with natural products, but what we've seen is throughout the intervention, we were clearly able to see differences in the patients. Just to give you a quick breakdown of what these patients look like, we specifically wanted to include people who were generally healthy before, right? Because if you have a sick population with long COVID, then we may make them better of long COVID, but they may still have heart disease or something. And so it just gets very messy. So we wanted to pick 10 people who were generally healthy. Strangely enough, the long COVID community that contacts us is fairly young. We see people in their teens to people in their 40s, maybe early 50s. That's really what we've been seeing. And so the average age here was 40. You had 5 females, 5 males. So, we see it across the board here. A few things we did. There were many different assessments that were conducted during this trial, but 2 measurements I think are going to be of interest to this group are we did a VAS score, which is a visual analog scale, and how it works is there's a 10 centimeter line. Okay, it doesn't have any numbers on it. A patient will have to mark on this line where they are in terms of that symptom. So when they're doing a VAS score for fatigue, for instance, you have fatigue or no fatigue, and you have to draw a line somewhere in there. The whole point of this exercise is that anytime a patient comes in to mark their assessment, because there's no number on this line, they shouldn't remember where they were before. So the idea is you're supposed to intuitively be able to kind of mark where you think you are in that given moment. And if you look at this, it's very interesting to see how it moves. You see a trend as they get their treatment. So there is a dose-dependent effect as the treatments are being administered. You also see their fatigue assessment score. That's another scale. That's not a visual scale, but that's a different scale where they do a self-reported outcome, like a survey, and we're able to measure that dramatically goes down as well. We do a lot of other autoimmune diseases. We've recently published on lupus as well, and we know that fatigue is sort of a general thing we'll measure across many studies, even our healthy populations. And when we see fatigue in any one of these trials, we have generally seen that resolve, fatigue dramatically resolve in many patients across many different indications. SF36 also has some measurements when it comes to fatigue, energy and fatigue, but also general health like sleep. Oved, you mentioned that. I don't have the exact scores for sleep here, but we can pull the data for all of that to see how it's affected. Parkinson's disease patients also have a lot of sleep issues. We've seen a lot of that resolve too in those studies. So it'll be interesting kind of to one day take all these data from different studies and maybe combine it to see what the overall effect is. But if you just look at these numbers, the fact that these patients were able to, pretty much all of them, get to a point where they are, their general health is all feeling better in the different aspects of their health, I think was really, it was eye-opening to us. First of all, from a success, we thought this is the most successful thing we've ever treated, most complex, because we actually don't have a real explanation other than immune system regulation. That was really it. But the fact that all these patients were feeling better during the course of this study was enough for us to now embark on a larger study. So we started a phase 2 double-blind, placebo-controlled trial for long COVID, which we are still recruiting. I think as of this morning, there are, 15 openings remaining. So this trial is going to be using allogeneic cells. Now, allogeneic means using a donor cell product. A donor, how we define a donor is FDA has these requirements of what a healthy donor is. And so we have to meet those requirements. It's a laundry list of things. And so we've selected our healthy donor, and this person's cells are going to be used across all 80 patients. Now it's randomized, so 40 patients are going to get the cells and 40 are not. But what we will be testing is a similar, this is the exclusion and inclusion, and all of this is available on clinicaltrials.gov, just so you guys are able to look that up after. But this is the protocol. Now, the upshot to this, unlike the first protocol, is because patients don't have to receive their own cells, there's no wait time. Once the patient is screened, they enter the study, and as soon as the first infusion is available, they can start. So you eliminate this 3-month wait. Plus, you eliminate, first of all clinical trials we conduct are free of charge. There's no cost to participate. But if you're in a trial where you want to use your own cells, then by definition, you have to be banked. Your cells have to be here in order for us to make the drug. So there's this cost involved in banking. This causes a lot of confusion sometimes because people are like, well, why do I have to pay? You're not paying for a trial, but we have to have your cells and you have to be commercially banked in order for us to be able to use them to make the drug. So in this instance, we don't have that. So it's completely, there is no cost involved at all. You don't have to bank, you don't have to do any of that. You just screen and you go into the trial. You receive 4 infusions. Why did we say 4? If you remember, the previous trial was 5 infusions. It was because by infusion 4, everyone was pretty much done and better, like to the point that they were normal as they were before they had long COVID, if that makes sense. So that's why we decided to cut it down to 4. This also helps the whole timing of the trial. The quicker we can get this done and reported, the quicker we can make this available to everyone. That's been the goal since day 1. And so that's the logic here with the intervention. You do see infusion 1 and 2, 2 weeks apart, and then rest 4 weeks apart. That is the same. And then you see in the follow-up, there's 3 follow-up visits. There are lab samples that are collected throughout. Those are safety labs to measure your, basic metabolic panels and just to make sure that everything is the same, which we have seen in every study remains the same. But for safety, that's what we're collecting. For biomarkers, people ask us all the time, are you collecting any other kind of blood test in association with the condition? If there were one, we would. So yes, inflammation is always there. However, it's not always there with every patient. And you can see that some patients might have very elevated inflammatory markers to begin with, and some may not have at all. And sometimes those people have worse symptoms. And it's very, from a biomarker standpoint, I don't think we're there yet. So we have to find one that, and I know that I think NIH is working on that. A few people are out there looking at those things. And so we're keeping close attention to those. We do measure inflammatory markers, particularly in conditions like rheumatoid arthritis and lupus and other things that are more consistent that we see across all patients. Long COVID has been challenging in that regard. So this is the overall schedule, and you'll see all of the assessments. On the left here is the list of all of the information that we will collect through it. And then, and hopefully we can report the study. Our goal is to get this study reported by the end of the year. That's what we hope we can do. So we can figure out what the next step is. And I think the next step, of course, going on to ME/CFS, I have personally been very interested in this condition. I think it's a great fit for the autoimmune applications that we've been doing. And when it comes to long COVID, I think we've really been thinking if our government is not interested in studying this in a larger way, then we will probably take this into ME/CFS. That's sort of the thought that we have. But with that, I'm going to leave it up to Oved to kick it off with questions.
Thank you, Donna. This was extremely interesting, and thank you for helping clarify many of the questions around the technology, the approach, and the study itself. Really appreciate it. The first question is, let's just focus on the ongoing study. I think we definitely, first of all, you described a very robust scientific approach to that. So I think we feel that this is a study that will really help us to get good answers. You mentioned that there are still 15 slots available. Could you say a few more words about what would be required from the people who are considering to participate in the study? Is that going to be in Texas only? Do the infusions need to take place there? Can you just say a few more things about that?
Yes. So the trial is a single site trial, and it's at our research foundation here in Sugar Land. So yes, people are traveling in for this trial. Again, given that the treatments and everything have to happen here, it will require, with screening, 5, 6, 7, 8 visits in total. So physically, you must show up here 8 times. That's a lot. It's a lot to commit to at the beginning. And another thing I want to touch on is, this trial I have seen has a higher dropout rate than other trials. And the reason I think is just given the data from the first trial, and you're seeing the response being so strong, I think, and now that patients are meeting each other and talking to each other in social networks, I think if they feel like they're not getting it, then they'll drop out. Okay, if they feel like they're in placebo, they're going to drop out. Let me tell you, if one does that, it affects the integrity of the entire trial, because if you have less placebos at the end, then you can't compare it against it adequately. So it threatens the overall study. And so when talking to people who are interested in traveling in, we are really hyper, just making sure that we're going to find patients that are going to commit to the travel schedule, no matter what group they find themselves in or they think they're in.
Yeah. And can you explain more about, there's some questions about immune response against the stem cells themselves. Is there any risk of developing antibodies or any immune response against the cells, particularly in the case of a donor? Stem cells are derived from cells that are not yours. Do you see any risk that's increased because of that? What is the experience telling us about that question?
So it's such a great question, and I have always been personally a proponent of using your own cells. I mean, if you think about it logically, if you were to ask anybody, do you want your cells or someone else's cells? The natural answer is always going to be, I want my own. But where is that coming from? Well, it's a thought that if I receive my own genetic material, I'm never going to reject it. So there is no possibility of me to develop something against my own stuff. Okay, makes perfect sense. So I've always thought, even though mesenchymal stem cells are shown to be immunomodulatory, and they have no HLA to be concerned of, which is, when you do a donor match, like if you were to get a bone marrow transplant, like cancer patients get bone marrow transplants, they have to match patients because of the antigen issue. But MSCs have low immunogenicity, so you don't have to worry about rejection. Now, from a scientific standpoint, every paper that you read will tell you it's actually perfectly safe, that there is no elevated antigens, nothing is happening. And clinically, we haven't seen it either. And so when we went into trials, I, again, was so committed to using one's own cells that if the possibility of rejection is there at some point, it could be, but we don't know. The answer is we don't know. When COVID hit, it changed everything, right? Because when COVID hit, suddenly, you're in a hospital ward and you're trying to help patients that don't have anything. They don't have cells banked. Majority of Americans don't have their cells banked somewhere. So that by default means that if you're in an emergency, you have a stroke or a heart attack and you're in a hospital, you're not going to have your cells banked. Then you have to weigh what's the risk and what's the benefit, right? And at that point, yes, receiving a cell product that someone's not going to reject based on all the scientific evidence for that it makes a lot of sense to save that person if you can with cells. So that's how we're kind of approaching this. Now, so far, I can tell you in our clinical trials, it has been so successful in terms of safety. Like I have seen no clinical signs of rejection, nothing from these patients that now every randomized trial we're doing, we run in parallel. We'll do an autologous group and we'll do an allogeneic group. So right now in Parkinson's, I have an autologous and an allogeneic. And for this too, we are doing the same thing. And I think what we are seeing so far in this long COVID seems to be the same, whether you're receiving a donor or whether you're receiving your own. Now, ironically, FDA, I think, has been used to seeing more applications using donor cells, right? Because since the beginning of bone marrow transplants, it's always been using someone else's. And so also the logic has been, well, if you're sick and you use a sick person's stem cells, it's probably not a good idea. It's probably good to use a healthy person. So if you see kind of where we've moved scientifically, it's very interesting, right?
Yeah. So maybe just to kind of wrap up the questions around the current study, if you could just maybe quickly let us know how long is an infusion? People are asking about that, and I think we will put in the chat the exact location to find the trial on clinicaltrials.gov, so it's easier for people. But if you could just say, how long is an infusion? And also the other question is for people who are going to be on the placebo arm, will there be an opportunity for them to receive the active drug at the end of the trial?
Okay, so this is what we are telling everybody. We have several studies that are randomized, and what we commit to is at the end of the trial, once everything is unblinded and all the study results are out, if the study proves that the treatment helped, then we will file an expanded access protocol to get all of the other people in. This is different than a traditional crossover design in a clinical trial. Crossover design by default means you're either in treatment and placebo, and halfway through the trial you switch. And then if you do this, your trial goes twice as long. So we're trying to not do that because the goal is to get this done and report. Yeah. And the infusion lasts 1 hour. And there is a 1 hour follow-up. So we'll just watch you for another hour. This, by the way, used to be 4 hours. FDA's now brought that down to an hour. So you only have to stay an extra hour. So it's 2 hours total. Probably add 30 minutes to see the doctor. So 2 hours and 30 minutes is your visit time.
That's great. That's very helpful. So maybe transition now to kind of the future a little bit. And we have started a conversation about the potential of using this approach that you just described so well to us in ME/CFS. So maybe if you could share your thoughts on why do you think this is, you mentioned that already, but why do you think specifically the areas that are really effective with ME the most, why do you feel that they could be benefiting from this approach? And then I think after your answer, we'll put a question to the audience. We're interested to see how many would be actually interested in participating in a study as you described. So, why do you think ME is a good disease to use mesenchymal stem cells as a therapeutic approach?
I see it because of it being autoimmune in nature. I think autoimmune diseases, there's like 900 different kind of autoimmune diseases. And if you look at the symptoms associated with all of these diseases, they vary greatly, right? That's why I think in long COVID, you have so many different expressions of the disease as well, because it can affect gastrointestinal, just that, like in Crohn's or in celiacs, or in like ME/CFS, it has to do with fatigue or post-exertion malaise, like those sorts of things, POTS disease, like all of those symptoms that we see. I feel like in the traditional pharmaceutical world, they've been trying to deal with the symptoms of autoimmune disease. And so the drugs are generally directed at trying to solve what is bothering the patient, which I appreciate. But if we can regulate the immune system as a cause for this chaos, I think we can, without even having to deal with the complex symptoms that all these people have, like in MS, if I think about the diseases that we're currently studying, if you're hyper focused on trying to mitigate the symptom response, that becomes very difficult. Yeah. But if we're just looking at the immune system, I tend to feel like once we're able to regulate the immune system, then inflammation tends to go down. And if systemic inflammation or neuroinflammation and neurological diseases, all of that comes down, then it at least gives your body a chance to recover, and it gives your body a chance to regenerate. Regenerative potential is greatly affected by inflammation in your body. So you have to deal with that. And I think that's why MSCs that are capable of regulating your inflammation, your immune system, and lowering your inflammation are going to be key in helping a lot of these autoimmune disease cases. And again, because I see fatigue in so many conditions, even in your healthy population, the older we get, we also suffer from fatigue. And so it's just something that I've seen across the board that I think would be really great to study in a specific CFS study. And I see one of the questions here, people asking if people with CFS are able to qualify for the study, and they are specifically not allowed to, because it has to be fatigue due to long COVID. That is what FDA is requiring.
So it will be interesting to see the results, obviously, in this group. So I think we're coming to the end of our time. Unfortunately, I think Donna would love to have you back, but maybe if we can, Carmen, if we can post the poll just for the audience, those of us on the webinar today that have ME/CFS that did not start with COVID. Would you be interested in participating in a stem cell therapy study, which probably would be along the lines of what Donna would just described? So if we can get people to provide their answers.
It says host and panelists can't vote, but I vote yes.
All right. So as we're waiting to see the results, Don, I think we would definitely like to continue the conversation. I think that and we can see that yeah, we have certainly a majority of participants sharing that they would be interested in participating in a study like that. So I think you've addressed the risks and the potential benefits, I think, in a very fair and balanced way. And so the response from the audience, I think, is telling us that this is something that we want to continue to work on. So, Donna, I wanted to really thank you for your time today. We will continue our conversation. For those of you who are in the Texas area or somewhere else where it makes sense for you to participate in the long COVID study, if you qualify, please contact the group directly. And I think helping the study to reach its recruitment target would be helpful for everyone to really understand the questions that we're asking. So again, thank you very much for being with us today, and I really appreciate it and look forward to more conversations. So Carmen, if we can ask you to help us wrap up the presentation for today.
Sure. Thanks everyone for joining us. I just wanted to let you know that this recording of the presentation will be available on YouTube in the coming days. And if you enjoyed this presentation and you'd like to help us continue to make them possible for everyone, we love to create free educational opportunities for the community. So maybe consider giving a donation to Solve to help fund our educational and outreach services. All right. Thanks everyone. Have a wonderful day.
Thank you.
Thank you very much.